When is a neural representation a conscious one? There are all sorts of devices that process visual information. A photo camera, for example, not only stores the images that you take, but it can also categorize part of that image as being a face. It can recognize that face and put a name tag below it, and it can even judge the emotional expression on that face so that it only takes a photograph when everybody smiles. Now, despite all that, we generally do not assume that photo cameras have conscious sensations of the photos they take. So why don't photo cameras have conscious sensations? The machinery inside the camera is fairly sophisticated. The CCD chip takes images at resolutions almost as high as the human eye, and the computations that go on in the face and smile recognition modules are in fact fairly complex. You could even argue that what goes on in the photo camera is very similar to what goes on in the human visual system, where you have the eye, where photoreceptors sample the image very much like the pixels in the CCD chip, where you have primary visual cortex, where neurons each look at very discrete parts of the visual field, and we too seem to have face recognition modules in the shape of the FFA, or emotional recognition modules, for example, in the shape of the amygdala. So why is it that when we look at a face we have a conscious sensation of that face, whereas when photo cameras take photos of faces they do not have conscious sensations of those faces? Now, to understand that, we looked at the difference between categorizing faces versus consciously seeing faces in the human brain using a technique called dichoptic masking. Now, in dichoptic masking you present two different images to the two eyes, but instead of having them rival, you make sure that these two images are fused into what is called a cyclopean percept. Now, we had faces in each eye, iconic faces defined by differences in the orientations of line segments, but these line segments were arranged such that when combined into the fused image they made up a homogeneous texture of identical crosses, so that the face which was present in each eye was no longer invisible in the fused image. And we contrasted that with a setup where the line segments of the two faces in each eye were arranged such that in the fused image a face was clearly visible. Using this technique we presented not only faces but also houses and other objects, nonsense objects in this case, which were then either visible or invisible to the subjects. We analyzed whether these faces evoked selective activity in the FFA by contrasting the signals from the faces with those of the houses and other objects, and indeed we found that these orientation-defined iconic faces evoked selective activity in the FFA. Now, surprisingly, it didn't really matter whether the faces were visible or invisible for this selective activation to occur. Both visible and invisible faces evoked significant selective activation of the FFA, both on the left and on the right side. So that allows us to conclude that both visible and invisible faces evoke very similar category-specific activation of the FFA. To further establish the similarity between the categorization responses for visible and invisible faces, we also used a classification analysis where we tried to classify the presence of faces from the pattern of activity in ventral cortex. And that showed that indeed you could classify the presentation of faces from that pattern of activity, but the classification could be performed equally well when you trained the classifier on visible faces and then classified invisible faces as the other way around, i.e. training the classifier on invisible faces and then trying to detect the presentation of visible faces. So that shows that indeed the pattern of activity, of category-specific activity, is very similar for faces that you either see or do not see. Now, of course, that very much begs the question what then the difference is between the neural activity that is evoked by faces that you see versus faces that you do not see. There must be some difference after all. Well, that difference emerged in the EEG responses that were evoked by these very same stimuli. Invisible phases evoke a EEG response that is very short-lived. You see a very short blip of activity in visual and temporal cortex that then very quickly dies out. Now for the visible phases you see a similar short blip of activity, but this is then followed by a much longer and much more extended pattern of activity that is probably maintained by recurrent interactions. And a similar difference can be observed in the frequency domain, in the sense that invisible phases do not evoke very much activity at all, whereas visible phases evoke prolonged activity in the theta, beta and gamma bands. And finally, we looked at the dynamic interactions between cortical regions using the fMRI signals. And there we observed that the visible condition is selectively characterized by much stronger and more elaborate interactions between the fusiform phase area and early visual areas like V1 and V2. So there is a clear difference in the way phases that you consciously see are processed compared to phases that you do not consciously see. Both evoke selective activation of feature detectors like orientation selective cells in primary visual cortex that respond to the line segments of those phases. And of course in both cases you have selective activation of the fusiform phase area, the FFA. But only in the case of visible phases, phases that you consciously see, you have interactions between these modules. So that may very well be the reason why in a photocamera there is no conscious sensation of the photo it takes. Because in a photocamera the pixels and all the modules do their job independently. They do not interact. Whereas in the human brain there is consciousness because the pixels and modules interact. The cells in early visual areas that detect local features interact with these cells in higher level category selective areas like the FFA. And of course this is very much related to the idea that consciousness is defined by what is called integrated information, an idea that was pushed forward by Giulio Tononi. Now let's have a closer look at what that integration of information, this interaction, actually means, for example, for cells in primary visual cortex. Cells in V1, primary visual cortex of the monkey, have very small receptive fields, so that they only look at a very small section of the whole image, a bit like the pixels in the CCD chip. So these cells are the ideal location to look for potential effects of the integration of information. And cells in V1 are typically also orientation selective, so when you present a few line segments within the receptive field as shown here of the appropriate orientation, they will typically respond. And you can then let these line segments be part of a homogeneous texture like shown here, where there's no specific object to be seen. Or you can have these very same line segments be part of a very circumscribed square figure as shown here. A figure that is defined by a difference in orientation between figure and background. You see the figure because you group all the line segments with the same orientation and segregate that region from the background, which has line segments of a different orientation. When you then record neural responses from such a cell, you will observe the following. Now from 40 milliseconds on, this cell will start to respond, but this response will be completely identical for the two stimuli, the figure and background stimulus. Indeed, as if the cell is only looking at what happens within its receptive field, which is of course identical in these two situations. So the cell is functioning completely independent of the visual context of the line segments within its receptive field, a bit like the pixel in a photocamera would do. However, from 100 milliseconds on, you will see that the cell will start to respond differently to the two stimuli. In this case, a larger response to these line segments when they belong to the figure than when they belong to the background, as if the activity is modulated by the global visual context of these line segments. So this cell is no longer working independently, but is now integrating information from what happens within its own receptive field with information from the surround. And this modulation is very much reflecting figure-ground segregation in the sense that the activity is elevated over all neurons that respond to the figure surface compared to cells that respond to the surround. And in fact, you actually see this modulation occurring slightly earlier at the boundary between figure and background, and then you see a sort of filling in for the figure surface after that. This modulation, reflecting information integration, is mediated by feedback from extrastriate areas back to primary visual cortex, because a lesion to these extrastriate areas abolishes this modulation, particularly in the center. The modulation at the boundaries seems to be mediated by more local interactions. In a large series of experiments, we have established that this contextual modulation, this signal of information integration, is related to consciousness in that it depends on the conscious state and also typically reflects conscious content. You can record from cells in primary visual cortex perfectly fine when animals are anesthetized, and cells will exhibit typical feature selective properties like orientation selectivity or direction selectivity. However, these kinds of modulations that I've shown here are completely abolished when the animals are anesthetized. We have also shown that when you mask these stimuli so that the animals are no longer capable of seeing them and detecting them, this also selectively disrupts these contextual modulation signals, whereas the orientation selective responses, as in the anesthesia experiment, remains intact. And this is very much in line with all sorts of experiments showing that when you apply TMS over primary visual cortex, at the moment that these feedback signals arrive, you also disrupt visual percepts, conscious visual percepts. And finally, we have shown that when you present these figures at various locations in the visual field and have monkeys report whether they see these figures or not, you will see that whenever they see the figure, you get this modulation. However, when they report not seeing the figure, the modulation is again selectively and completely absent. So this shows that recurrent interactions, feedback interactions between neurons in visual areas, produce conscious visual percepts as they integrate information from low-level and high-level feature detectors. And this shows that this only happens in the conscious state, as this does not happen in anesthesia. And it also shows that interfering with these recurrent interactions, as in masking or TMS, disrupts conscious percepts. Now, I would also like to add some notes on the molecular mechanisms of these interactions. In this more recent experiment, we again showed these textured figure ground stimuli and recorded from neurons in V1, but now also simultaneously injected drugs in primary visual cortex that selectively blocked specific neurotransmitter pathways. And what we then found was that applying APV, which is a selective blocker of the NMDA receptor, interferes with the recurrent processing signal, with the contextual modulation, as you can see when you compare the blue with the red graphs or the bar charts. Which of course strongly suggests that the feedback which is mediating these recurrent signals is processed via the NMDA receptor. And what we also found was that applying CNQX, which is a selective blocker of the AMPA receptor, this strongly reduces the feed-forward visual signal itself, however without tampering with the recurrent signal. The role of the NMDA receptor in feature integration was then confirmed in human subjects where we applied ketamine at a subanesthetic dose, a dose at which ketamine primarily acts as a NMDA receptor inhibitor. And what we found was that by applying ketamine, you again selectively interfere with the ability of these subjects to detect orientation-defined figures. Another interesting effect of ketamine was found when we compared the patterns of activity evoked by different Mooney images. Now, Mooney images, as you may know, are rather difficult to identify, to recognize when you've never seen them before. But once you have seen the grayscale version of that Mooney image, you're typically much better at recognizing the Mooney image. What we observed was that before recognition, the patterns of activity that were evoked by these different Mooney images were rather similar. What you have here are what are called dissimilarity matrices. And what you can see is that the dissimilarity is rather low between these different Mooney images, sort of indicated that from a neural perspective, these Mooney images more or less all look the same. Now, once you have seen the grayscale version and then look again at the Mooney image so that you recognize it, you will see that the neural patterns of activity are much more dissimilar. So from a neural perspective, the brain distinguishes better between these different Mooney images, like the brain does with the grayscale images. Now, what we observed in primary visual cortex was that this effect of recognition, that the Mooney images become more dissimilar once you recognize them, does not occur when ketamine is applied to these subjects. And moreover, this effect of ketamine on the patterns not becoming more dissimilar was particularly strong in early visual areas, in V1 and V2, and was not that strong in higher level visual areas. So this again suggests that the conscious recognition of, in this case, these Mooney images depends on the NMDA receptor mediated feedback from higher visual areas to lower visual areas. So another conclusion is that these recurrent interactions between neurons that produce conscious visual percepts seem to selectively employ the NMDA receptor pathways. And of course that suggests a particular link between consciousness on the one hand and the ability to learn from visual input on the other end. As of course we know that the NMDA receptor is specifically involved in learning and synaptic plasticity. And that in turn may be another reason why a photo camera does not have conscious sensations, as of course a photo camera does not learn from the images it takes, whereas we do. A final question I would like to address is whether these recurrent interactions, this integration of information, requires higher level functions, such as attention, or the ability to access and report the visual information, as is, for example, proposed in global workspace theory. Do you require these higher level functions to have a conscious percept, to have integrated information? It does not seem to be the case that this integration of information depends on attention, because contextual modulation, as you can record in monkey primary visual cortex, does not depend on attention, because it is equally strong per object, where the monkeys have to simultaneously attend either 1, 2, or 4 figures in a task where they have to monitor these figures for a change in the orientation of these figures, a so-called change detection paradigm. In fact, the modulation is equally strong for these 3 different conditions, attending 1, 2, or 4 objects. In human subjects, we used an inattentional blindness paradigm. And that entails that subjects have to very strongly focus and attend to a central letter stream, where letters are constantly presented at a rate of 6 per second. And then every now and then there's a white letter presented, and they have to indicate whether that letter is a vowel or a consonant. So that is a highly demanding task. And simultaneously we present textures, images in the surround of these letters. Now every now and then with this white letter there is either a homogeneous texture or a texture in which you can identify, in which you can see these figure ground segregation stimuli. And then afterwards we ask subjects whether they had noticed the presence of these checkerboard figure ground stimuli. And what we observed is that a lot of subjects do not notice these checkerboards at all, indicating that they suffer from what is called inattentional blindness. They have not seen these checkerboard textures, and they were unable to report them because they had no access to this information while they were focusing on the central letter stream. And remarkably, regardless of whether subjects notice these background textures or not, and as is the case in inattentional blindness, the signal reflecting the integration of these elements, the contextual modulation signal, is exactly the same for the situation where subjects suffer from inattentional blindness, i.e. do not notice these background checkerboards, compared to the non-inattentional blind situation where subjects are aware of these background textures. And in a final experiment, we looked at perceptual integration from an entirely different perspective, as you can observe in what is called the Canissa illusion. Of course, in the Canissa illusion, you also have to integrate information, in this case from the different Pac-Man inducers, leading to what is called perceptual inference, where you can observe what is an illusory contour and importantly also an illusory brightness increase of the surface in between these Pac-Man inducers. And importantly, it has been shown that this Canissa illusion is not perceived when the stimulus is made invisible by masking. So apparently the inference that you get, the integration of information leading up to the perception of the Canissa illusion, seems to depend on consciousness. We combined the Canissa illusion with the inattentional blindness paradigm by having subjects again focus on a central letter stream and doing a particular task with those letters, while we presented in the surround these inducers. Now we had 4 different types of inducers. The one which induced the classical Canissa pentagon in this case, a Canissa out control where the inducers were pointing outwards so that you do not have the Canissa illusion. And we also had 2 other controls, lines out and lines, where you have a similar pentagonal configuration of line elements but without the strong Canissa percept, but in particular without the illusory brightness induction that you have in the Canissa illusion. Now afterwards we asked subjects whether they had noticed the presence of the Canissa and other configurations, and subjects who reported not having noticed this were called inattentionally blind, and we then compared their neural responses to the neural responses of those same subjects after they were pointed out that these Canissa pentagons were present. What we observed was that the Canissa pentagon evoked a stronger pattern of activity over a set of visual areas than the control stimuli, indicating that the Canissa evoked specific neural signals. And what we also saw was that these Canissa specific neural signals are present both during the inattentional blindness condition and the non-inattentional blindness condition, indicating that you don't need attention, access and reportability for the feature integration, for the information integration leading up to this Canissa illusion and leading up to what is called inference. So that allows us to conclude that the interactions leading to feature integration, perceptual organization and inference also occur in the full absence of attention, access and report. And therefore it actually is a mistake to conflate visual consciousness with attention, access or report, as is done, for example, in global workspace theory. So, in conclusion, recurrent interactions between neurons in visual areas produce conscious visual percepts as they integrate information from low and high level feature detectors. And at the single neuron level, in V1 for example, this is reflected in what is called contextual modulation, which is absent in anesthesia, masking, or when you apply TMS, or when monkeys report not seeing these figures, which strengthens the relation between these information integration signals and consciousness. Now, moreover, these interactions selectively employ the NMDA receptor pathways, which strongly suggest a link between consciousness on the one hand and learning on the other. And finally, these signals reflecting information integration do not depend on attention, access or report. And hence, we should conclude that consciousness is present without attention, access or report. I would very much like to thank all the people who have collaborated on these experiments, either in the past or more recently or currently. People like Hans Super, Rogier Landman, Johannes Varenvoort, who have now left the lab, Steven Scholten, Matthew Self, who is working with Pieter Rolsema, Martijn Wokke, Julia Meuse, Anouk van Loon, and Annelinde van der Broeke. Thank you very much.